Okay, so good afternoon or good morning, everyone, depending on the place where you are. I'm Soneme from Sansusi Research Scientific in Côte d'Ivoire. It's my pleasure to welcome each of you to this writing workshop. So on behalf of the African Bioimaging Consortium, I'm glad to give that introductory remarks and talk to you a little bit about the consortium. So as you can see, ABIC, African Bioimaging Consortium, is a community-driven initiative that wants to empower the African bioimaging and microscopy community, and they aim to unite and grow African microscopy community, promote microscopy in research, connect scientists, build dialogue and empower advocacy. And it is an open and free consortium, open to all career stages, all expertise and fields. It's also open to international and commercial partners. Here is some of the things you can see on our website, africanbioimaging.org. And I want to invite you to visit that website and pay attention to some of the opportunities and travel fellowship we are having there. From there, you can also sign up for our new letters, add your information, and join one of the 3 working groups we have about training and education, growing imaging infrastructure in Africa, and communication and awareness. You have also the possibility to write a blog and join the community meeting. So scanning this QR, you can connect, and I want to highlight our upcoming online community update meeting, which will be held on April 20th at 1 p.m. UTC. So you can register and attend the community meeting. I think maybe in a few minutes, Karen can talk a little bit about this. For this application, the whole process is the following. So for today, we have a workshop. There will be another one in April 20th. Then we'll follow the clinics, writing clinics, and the submission. Kindly note that the deadline is May 29th. In this workshop, you will have to interact with a couple of people from the Africa Microscopy Initiative. So Michael will give a short talk on this. Then from ABIC, myself and Karen Jacob, and you have a full team from the Advanced Imaging Center who will support you through the process. So thank you for your attention. And there is a link. So Karen, I will let you the floor for this link about people to tell us where they are from.
Sure. Thanks, Sameh. And it's great to see everyone here today. I'm just pulling up the deck, one moment. So we have, when we asked you to register today, we just asked for your email address really, but we would love to know where you're all joining us from today. And we also would love some information from you on how to plan future ABIC writing workshops. So I hope you're all seeing a screen with a QR code. If you go to use that QR code or follow the link that I've just put in the chat, there are 2 questions that it would be really great if you could answer quickly in this workshop. I'm going to leave this QR code up for a few more seconds before I go to the next slide. So firstly, we'd love to know where you're joining us from today. So please feel free to just, as you go to menti.com and enter in that information, if anyone's having problems with Menti, just stick your hand maybe at this stage. But it would be great to see where everyone is joining us today.
He's asking for
One
password. Password.
Mike has just let me know you have to go to menti.com and either enter this code, this 5833010, or just use your mobile phone and follow the QR code. While everyone answers that, I'm going to move on to the next question, which we can return to at the end of this workshop, which, as Ame said, this is the first of a series of workshops that ABIC wants to host, helping the African community in writing applications for workshops or meetings that they want to attend. So while this workshop is for the applications for the Imaging Imagery Center, let us know what other specific workshops or meetings or funding calls you would want us to arrange workshops for. And that helps us plan future workshops. So you can think about it. If you can't think of anything today, please send us an email. I may have the ABIC information up so that we know what workshops to plan in the future. For now, apparently we still only have people from South Africa and Uganda here today. Perhaps we can move on for now. I don't want to spend too much time here, but if people can access the Menti and let us know about the workshops, that's the most important thing. So I'm going to stop sharing and hand over back to you, Ame.
So thank you, Carol. The next step of the program is to give the floor to Michael. Hey, Mike.
Yep, I'm ready. Thanks, Amir. And then quickly, thanks to both ABIC and the AIC for hosting this writing workshop. I think it's a really great idea. So what I'm going to do is I'm going to quickly just spend 5 minutes giving a brief overview of AMI and then the Visiting Researcher Program, which I'm sure all of you are familiar with, which is why you're at this little writing workshop. So I just want to confirm if my screen is being shared. Cool, great. Okay, so a brief, quick overview of the whole of AMI. So AMI is a large kind of initiative, the African Microscopy Initiative, that involves multiple different things that are being pursued. One of them is the Imaging Center, which I'll talk a bit more about. There's also got a good link with the Imaging Africa workshop, which was started at the Advanced Imaging Center, but is now kind of being assimilated into the African Microscopy Initiative. And this is the all expenses paid microscopy workshop that was hosted in 2020. And then again, there was one last year. There'll be one towards the end of this year or the beginning of next year. There's also the Microscopy Matters webinar series, which is still a work in progress that AMI is working towards. There's the PITCH program, which is Partners in Teaching, and this is a program that will see African or aspiring African imaging scientists that are interested in microscopy and imaging related sciences, pairing up with another researcher from elsewhere in the world to host a microscopy workshop at the African Researchers Home Institute. So this is quite a novel program, and it's still a work in progress. So this should be announced either end of this year or beginning of next year once again. And then the last component is PEER, which is an equipment redistribution program that we're also working on. So the last three components are still a work in progress, as I mentioned, but I'm not going to discuss them. If you do want more information, you can either reach out to me or take a look at the website. But for today's quick short presentation, I'm going to be focusing on the AMI Imaging Center and then its visiting researcher program. And specifically, I'm just going to be covering three broad topics. The visiting researcher program and what it is and what it kind of entails. I'm going to give you a little bit of an insight into the application process to hopefully contextualize why the application is so important and why something like this writing workshop is actually a really good opportunity for you to write the best application, because it's quite important down the line. And then at the end, just as a bit of not necessarily fun, but I'm going to lay a challenge to everyone that's attending this workshop at the end of my short talk. Oops, sorry, that was a mistake. All right, so we'll go into the visiting researcher program first. And essentially what the visiting researcher program is trying to do is it's trying to make imaging and optical fluorescence microscopy and transmitted light microscopy technologies accessible to researchers in Africa, because there's incredible things that can be done with microscopy, but unfortunately these technologies are often expensive. The expertise required to perform them is not necessarily widely available. So what the African Microscopy Initiative is trying to do is we're trying to make instruments available for free for researchers to come and do work on these microscopes that is relevant to the people of Africa. So, you know, malaria, infectious disease research, neuroscience, whatever it might be, we want to be able to accommodate all sorts of science at the Africa Microscopy Initiative's Imaging Center, which is hosted at the University of Cape Town. So this is a wide angle photo of our lab. It's got a lot of equipment in it. I'm not going to spend that much time going over what we have because it's actually very exciting and I would talk for hours about it all. But essentially we have 5 different microscopes that have a variety of different uses and use applications that I'm offering and we're extending to you, researchers throughout Africa, to apply for free to use. So as I mentioned, there's the 5 instruments. They have a wide variety of different applications and all sorts of technicalities that are far better suited to be discussed over on a one-on-one discussion. So I will say, if you do want to talk about what is available, what can be done on our instruments, I ask you to get in touch with me and we can set up a time to discuss. And but I'll get more in. I'll talk about this more towards the end. But essentially, what this means is that we want to be able to empower researchers in Africa to bring whatever samples that they need, and we can find a way to extract useful and powerful information using microscopy, which is something that's not easily attainable because it's just so expensive usually and expertise unlimited. So this is quite literally a unique opportunity. So I hope everyone is excited by it. But it's not only about the microscopes. What we offer at the Army Imaging Center is a whole entire package, and this includes a biosafety level 2 facility. So there's space there to do cell culture if you need to do that. There's space and equipment for sample preparation. There's general lab equipment. We'll provide image data analysis and support, dedicated assistance on the microscopes for around up to about 4 weeks at the lab. It also includes manuscript preparation and support. As you probably know, we will pay the accommodation of visitors to the lab, as well as the air travels to come to Cape Town. And then we also offer opportunities to get in touch with global partners from across the world to the benefit of your research. So I want to kind of highlight why the proposal is so important and why this writing workshop is so good. And I'm sorry, this text is quite small, but you don't need to be able to read it. Essentially, there's an expectation that if you come for 4 weeks, you're going to be doing sample preparation, microscopy, and image analysis. And there's a general expectation that this will flow in a linear kind of trajectory where one day you'll work on one thing and it'll all work perfectly and everything will be happy. But unfortunately, this doesn't always happen. And it's the reality of microscopy, and it's the reality of a lot of science, that we end up needing to troubleshoot along the way. And this is why the application is important, because it helps us identify potential issues that might be challenging, and it allows us to prepare in advance before you come to the lab, so that we're able to ensure that by the time you leave the lab, you're able to leave with a perfect data set that has all the information that you could possibly need from it. So about the application process. So we'll talk about this now. And essentially, you'll know that this needs to be done online. And part of the proposal, or rather the application, is the project proposal. Now, the project proposal is the most important part of the application. And there's information online. But again, if you want to talk about this, please feel free to contact me. But essentially, it needs to contain 7 parts. Firstly, a project summary, which just gives an overview of the work that wants to be performed at the Army Imaging Center, a background as to why this is important, a very clear aim and hypothesis that is kind of the driving factor behind this entire proposal. Insight into experimental design. You don't have to know exactly how everything is going to work, but it's a good idea to have an understanding of how certain milestones or how the pipeline is going to be achieved while you're in the lab. And this includes having an idea for what kind of data you want to be able to extract from those images that you end up taking. It's important to highlight the justification for why you're writing a proposal to the Army Imaging Center, mainly on a technical perspective, but also on a personal and accessibility perspective, because these instruments are not readily available. So it's important to highlight that. And then, of course, the impact of the work that you'll be doing. The review process of the proposals is very simple. It's a 2-tiered system. There's a first internal review, which covers technical feasibility and sample suitability or safety. And if both of those are approved, it then moves over to a global review panel where the scientific merit of the work being done and the justification for the experiment gets assessed by a group of international researchers. And it's quite good to contrast that the first internal review, you can get a lot of information on how to provide the best information from me during a technical consult. But this writing workshop is a very excellent way of making sure you can provide the best scientific merit and the justification for the eventual review by the global panel. Quick overview of the application. So hopefully you would have planned and designed in discussion with us, and by attending these workshops, you'll submit your proposal. It'll be reviewed by the panel. And then once it's been accepted, hopefully we will prepare for your visit and we'll start getting things ready for your visit, which would likely be about 6 months after the case. where you would come to Cape Town and we'd start doing these experiments. And then my last very quick challenge to you, to all the attendees of this writing workshop, is that microscopes allow us to observe our samples in new and exciting ways, but it can do so much more than simply allow us to see, okay? A well-designed experiment can provide information on the mechanism behind the biology that you're studying. So my challenge to everyone at this writing workshop is to see how you can take this workshop and consult, if you get in touch with me, and see how far we can push your research to perhaps perform some experiments you've never even thought of, right? This is an opportunity to use new tools and to think outside of the box. So my simple challenge is, let your imagination go wild. Who knows, you might want to end up submitting 2 proposals, one normal proposal and then one proposal that's a little bit more exciting and adventurous. So in conclusion, I will just finish off by showing this final slide where you can scan this QR code if you'd like to schedule a consult with myself. Otherwise, please feel free to email the link provided there, and we can get in touch to discuss your work, and I can tell you more about the microscopes. And then I'll quickly thank the Bill and Melinda Gates Foundation, the Chan Zuckerberg Initiative, and then the University of Cape Town for supporting AMI.
Thank you, Mike. So we noticed that on Menti, we have very few persons who answered. If the link is still open, Karen, maybe people can still join in. But our next step is to go now deep on the things and let the floor to the IEC team led by Prof. Leon. So Prof. Leon.
Thank you. Can everybody hear me okay?
Yeah.
Great. Good afternoon, everyone. It's very nice to see everyone here. And then we also have the entire AIC team, as you can see in one corner of your screen. They will chime in whenever they feel like chiming in. So I'm going to share my screen now. So before I share my screen, I sort of want to give you a taste of what is coming. So as a reviewer sitting on both Tier 1 and Tier 2 of the Advanced Imaging Center here at Janelia, we review a lot of proposals. And so a lot of this application process at the AMI Imaging Center is modeled after the AIC review process. And what I'm going to share with you is how, as a reviewer, we will evaluate your proposal. If you have not read the application website of the Imaging Center carefully, Mike has written a very nice instruction, and I suggest that you read it very carefully after this workshop, because the entire workshop is based on what Mike has written. So the very first thing about submitting an application is to read the instruction very carefully and read what the reviewer will be evaluating your proposal. So I'm going to share my screen now. Can everybody see it okay? Good. Everybody see it okay?
Yeah, all good.
All right.
Yeah.
Okay.
Okay. Right. Thank you. So these are the various components of your proposal. So there's project summary and the background of your research, and then the specific aims, hypothesis, followed by experimental approach and design, measurement and quantifiable outcomes, the justification for the microscope use at the AMI Imaging Center, and the impact on you and your laboratory. Every single section of this 7 components will be scored. Some will be weighted more heavily than others, and we will discuss this as we go along. So I'm going to go through this various components one by one, just so you are aware of what you are required to write and how we will evaluate your proposal. Project summary and background are things that you know the best. In fact, you should know more than everybody else because this is your project, right? So a project summary is just equivalent to an abstract of a poster or a paper that you are about to write. It's very short, hopefully about 100 words or so. And it very quickly let the reviewer know what this project is all about. So keep it very brief because you want to save the space for things that matter the most, which is the next few sections. And then your background, again, just like the background or introduction that you would write for a paper. In this section, you will describe the key unanswered questions. And this is very important because it actually helps you frame your project. Where does your project fit in that particular field of research that you are doing? What angle are you trying to tackle? Every field always have unanswered questions, right? So where is this niche of your project fitting into this entire field? And tell us a little bit why addressing this particular question is scientifically important. And describe the system that you will use to address these questions. Are you going to be using tissue slides secured from animal, from patients? Are you using Drosophila? Are you using any kind of organ explants? Or if you're working on ecological samples, is this sample collected from water reservoir or from the soil in the farm or whatever it is? Tell us the kind of samples that you'll be using to address this particular questions and why this kind of sample matters. And again, keep it brief because these two is just sort of an introduction for the reviewer. So while it will be scored, it will not be scored very heavily. Now, as you can imagine, if you go back to the 7 points that the proposal requires you to write, the next 3 are the ones that the reviewer will really score you. These 3 will make or break your proposal. So we are going to spend a little bit more time talking about these 3 components. And by the way, hang on. By the way, everything that I shared today will be shared with Amy, and so that Amy can help distribute it, so you don't have to take a lot of notes. And the recording will also be shared as well. All right, I have a question somewhere, right?
I was just going to chime in with one thing on the summary and background. It can be really helpful at times just to include a simple schematic to help the reviewer get introduced to your system, especially if it's something that's not very commonly used. So, remember that your reviewer is not an expert in your system. And so if you can make it really easy on the reviewer to understand what your project is, where you're coming from, sometimes a schematic or a simple drawing is a really easy way to help with that.
Thank you, Chad. That's a good point. Any questions at this point? And feel free to interrupt me anytime you want. You don't have to wait till the end of the presentation to ask me any questions. All right, if there is no question, we are going to go into the specific aims and hypothesis, experimental approach and design, followed by image analysis. So the very first thing you have to think about, and this is a paper that Michael and I have written a couple of years ago, and Mike, can you make sure that you share the PDF with Amy so that he can distribute this as well? And you will see this diagram in the figure 1 of the paper. So again, you don't have to take a screenshot or anything. You will actually get the entire PDF. Now, if you look at panel A at the very top row, basically, I'm going to see if I can get a laser pointer. Can everybody see my laser pointer? Okay, so yes. Usually, when you are doing an experiment, you will have a hypothesis, you plan your experiment, and then you prepare your sample, and then you go to the microscope, you take a few images, and then you process and you analyze. Sometimes you need to repeat it, but at the end, you do your results data presentation, either as a paper, at a meeting, or on a poster, right? So this is a very forward flow of an experiment. But if you look at the experimental planning phase is pretty much your proposal writing phase, because that is how you plan your experiment. The flow is actually in reverse. So this is this second row is pretty much the essence of what I'm going to tell you today, is that you already have a hypothesis, and your hypothesis has to be very focused, and it has to be experimentally testable. It is very important you frame your hypothesis that way, and I will spend a few more minutes in the next few slides to show you why that is important. Once you have your hypothesis, the very next things you have to think about is what kind of results will be informative so that you can use it to challenge that hypothesis. So if you don't have a hypothesis, everything else falls apart. So once you have an hypothesis, you can determine what kind of informative results you require. And once you know the kind of informative results that you need, then it will actually help guide you to the kind of data you need to generate to produce that informative results, right? What kind of data give you the results that you will need to challenge the hypothesis? And in addition to that, what kind of variables, experimental variables, do you need to measure, such as fluorescent intensity, cell density, direction of a particle movement, the velocity of a particle movement, or whatever, that will really require to give you the information you need to challenge the hypothesis. Knowing the kind of required data will actually help the AMI staff, such as Michael, such as Karen, to help you select the right kind of parameters. How fast do you need to image this sample? What kind of fluorescent channels do you need? How deep do you need? What kind of other imaging parameters that is required for you to help generate the required data to give you the information to challenge the hypothesis. And from there, the AMI staff will help guide you to the right microscope that you will need for the entire workflow to work perfectly. So you will notice that the entire information process goes in reverse. And a lot of people are under the impression that once you have a hypothesis, you need to select a microscope, which is totally untrue. Microscope selection is, in fact, the very last step of the process. And so hopefully with this flowchart, you will now realize that the most important thing is hypothesis formulation, okay? And the rest, I'll leave it up to you to explore in the paper. And I strongly encourage you to read that paper because it actually helps you quite a bit before you sit down with Michael and Karen for your technical consult. Okay, so hypothesis. I know that everybody have learned about scientific hypothesis in your high school, in your undergraduate, that science is usually driven by hypothesis, but it is very surprising that a lot of people don't know how to write hypothesis carefully. So the very first thing you need is to go back to your introduction section or your background section, look at the central questions that you have written, right? In the previous section, we talked a little bit about central questions. Now think about how you can translate that central questions into a hypothetical statement. A hypothesis does not have a question mark. It is a statement, and it is a statement that can be challenged. And so always think about how you can do that translation in a very rigorous way so that your central questions now turn into a hypothetical statement. And then phrase your hypothesis in such a way that it can be experimentally tested. And a lot of time, even in our own experience when we're dealing with scientists from all over the world at the Advanced Imaging Center at Janelia, and we realize that a lot of biologists are very, very protective of their hypothesis, as if their hypothesis is wrong, then they are a bad scientist. That is not true. Hypothesis is to be challenged. Even if your hypothesis is wrong, you learn something very, very valuable. And that is the whole point of the hypothesis. So don't be protective of your hypothesis. Instead, you want to challenge it as rigorously as possible using the experiment that you are going to design. And a hypothesis that is very focused actually help focus your proposal. And a focused proposal always score much higher in the eyes of the reviewer than a very, very vague proposal that have a hypothesis that is very vaguely stated as well. So if you look at the hypothesis 1 and hypothesis 2, for example, Hypothesis 1 says that we hypothesize that chronic inflammation may be involved in driving tissue damage commonly observed in disease X. That is not a wrong hypothesis. Many people write hypotheses that way. However, that is less focused than a hypothesis 2. We hypothesize that disease X leads to increased density of CD8 positive cells in the affected tissues. So even though both of them pretty much address the same thing, by focusing your hypothesis into something that can be experimentally tested, because hypothesis 2, as shown on your screen, can be easily tested. All you need to measure is the density of CD8 positive cells in the various tissues. So that immediately becomes a more rigorous hypothesis because it can be challenged by an experiment. Whereas vague words like involved or participate are words that cannot be rigorously tested in an experiment, because very few experiments can really test whether or not something is involved. In fact, even if you do a knockout, if you do a mutant, you say whether or not you're actually testing whether or not that particular event is required or that particular event is dependent on something else. It still doesn't test whether or not something is involved. So vague words like involve, participate are usually words that should be avoided when you formulate a hypothesis. Okay, so keep your hypothesis focused, and that will actually help Army staff when they are trying to assist you in writing a proposal. So keep it, and this is completely up to you. It is completely on you because nobody can actually help you formulate a hypothesis. This is your science. It is not the science of Michael. It is not the science of Karen. So you will be the one responsible for coming up with a hypothesis that is rigorous. Any questions at this point? Is there anything in the chat room? Because I cannot see it very well. If there is anything in the chat room, stop me and somebody read it for me.
Hello?
Yes.
Okay.
Is there a question?
Equal to the format.
No, Prof. It is someone who is having a call. Let me mute it.
Okay. Should I continue?
Yeah.
That was what I said.
Okay.
You have to put questions.
Wait, what was that? Sure, I'll continue. All right. So with your hypothesis clearly stated, now you can actually go into the specific aims. In the specific aims section, you should articulate very, very carefully and very specifically the kind of experiments that you would like to perform. And the experiment that you design, if your hypothesis is stated correctly, then the experiment that you design must be designed in such a way that it can challenge the hypothesis. And that is very important. We have also seen many, many times in our own experience where the hypothesis is here, the experiment is there, the experiment has no relationship with the hypothesis whatsoever. So that is a bad example of writing a proposal. You should never write your proposal that way, where your experiment has no relationship with the hypothesis whatsoever. It is also good to state the predicted outcome that could support your hypothesis. If your hypothesis is correct, then this is the predicted outcome. If the outcome did not turn out the way as you predicted, then your hypothesis is wrong. It may tell you something actually quite valuable, and then you can always retool your hypothesis and come back again. But you got to have a hypothesis that you can test, right? Now, each hypothesis must have its own set of experiments. It could be one single experiment or a combination of 2 or 3 experiments that can address that hypothesis. You cannot have a single hypothesis and have like 8 or 9 different experiments. Only 1 or 2 of them are related to the hypothesis.
Yeah.
If you have 2 smaller hypotheses, then you can create specific aim 1, specific aim 2, and each specific aims has its own little shorter hypothesis followed by a specific set of experiments. Question?
Yeah, someone is asking on the chat, is there any way we can be sure our hypothesis is focused enough?
Okay, example 1 and example 2 below are a good example. So if you look at example 1, actually this should be example 2, not hypothesis 2. I'll change that before I share the slide. Example 1 is somebody write a specific aim, say we will characterize the tissue samples from patients with disease X. That is a very unspecific specific aims. This is another words that you should absolutely avoid when you're writing a proposal. Does anybody know what this sentence means? It doesn't mean anything. You're just going to characterize it. All right, you take a picture. Now the picture is staring back at you. How are you going to characterize this tissue? Nobody knows. And as a reviewer, I certainly don't know what you mean by that, right? But if you look at example 2, it will say we aim to compare the CD8 positive cell population density in tissues collected for patients with disease X and compare it with a normal control. If our hypothesis was correct, we would expect to see an elevated CD8 positive cell density in the disease X tissue compared to control. Now it becomes very specific, right? So both of them, in a way, in the mind of the applicant, they are doing the exact same thing, is that the second applicant write it out very carefully so that the reviewer can actually understand what you're trying to do. When you are writing a proposal, when you are writing a grant or anything, you always put yourself in the shoes of the reviewer. If you write a sentence and then you pretend that you are the reviewer, can the reviewer actually understand what you're trying to say? Can the reviewer score it? Or the reviewer think this is going to be too vague? And if you, as your pretend reviewer, don't like that sentence, then don't write that sentence. So it is that simple. Okay. Anybody from the AIC team want to chime in anything? Or anything?
Yeah, I wanted to chime in real quick, just to add to the question that Mai mentioned, and I'll echo Leong. This is incredibly important part of the proposal, but this is also something that a technical consultation can help with. If you're having trouble articulating a hypothesis that you think is specific enough, talk to Mike, talk to Karen, talk to them, and they can help you refine your hypothesis to the point where it can be tested at a microscope. So I just want to make sure that everybody knows that.
And I'll add one other thing. When you're thinking about, how focused should it be, keep in mind that amount of time that you would spend at the imaging center, right? You're going to be there for 4 weeks, so a month. That's, a couple experiments that you're going to be able to do. It's not going to be kind of a 2-year plan. So really think about what can I get done in 1 month, and try and scale your hypothesis to that amount of time that you could spend in a lab, almost as another way of thinking about it.
Adding on to what Chad says, depending on how your hypothesis is framed, you would have to design your experiment. If your hypothesis is extremely vague and you image it without the optimum parameters, and then you try to refine hypothesis data, you may not have the data to be able to test that hypothesis.
And I want to also add to what Jesse says. Michael and Karen can certainly help you solidify your hypothesis a little bit more, but I don't want anyone to use them as a crutch and just go there without a hypothesis and say, all right, this is what I want to do. Can you help me with a hypothesis? That is a wrong approach. And more importantly, you put a lot of burden on Michael and you put a lot of that burden on Karen. What we would like to see is you try your very best to come up with a hypothesis that you think is rigorous enough, and then ask Michael and Karen for their opinion. And if they don't think it is good enough, then they can help you improve it. You have to do most of the legwork. They are there to help you improve your proposal. They are not there to help you write the entire proposal from scratch. So do most of the thinking. In fact, 90% of the thinking and consideration should be done by you, not by Karen, not by Michael. Okay, this is very important. They are there to help you. They are not there to design your entire experiment from scratch. Karen, you raise your hand.
I'm just going to jump in quickly here myself, and I will tell everyone what I'm going to tell you if you schedule a consultation. I expect your hypothesis to tell me what you are looking for at the microscope. Characterizing ABC doesn't tell me what you're looking for. Saying upregulation of a gene or a protein tells me you're looking for signal intensity. Stimulation of cell proliferation tells me you're counting cells. Increased cell motility tells me you're looking for cell movement. That's the sort of stuff I'm looking for. So if you read your hypothesis and says this tells the person what I'm looking for at the microscope, then your hypothesis is probably focused enough.
Yeah. Sunday.
Thank you. Thank you. My question is on expected outcome. Can you hear me?
Yes.
The question is, in a situation whereby you are going to be making use of some equipment that you have never used before in your previous studies, how do you overcome the challenge of expected outcome, because you have never used that equipment before, and how do you overcome such? I would like your comment on that.
So that is a good question, and in a way that ties very closely to what Karen has just said. You should expect what kind of questions, or let me put it this way, what kind of variables will challenge the hypothesis. Again, if you're looking at gene expression, if something is expressed, the intensity has got to go up. No matter what microscope you're using, every microscope can measure intensity value. Am I right?
Yes.
Yes. So that is all we need. You should expect that if my hypothesis were correct, then the intensity of this gene, the intensity of this protein should increase. No matter whether it is a confocal microscope or a simple wide field microscope, all the way to something as complicated as a lattice light sheet microscope, it doesn't change the fact that intensity will go up. And all you need to say is that we expect that intensity will go up. Now, some microscope will measure it better. Some microscope will measure it in a different angle, whether it's live cells, fixed cells, super resolution, deep imaging, doesn't matter. You are still measuring intensity. So it is actually simpler than you think. I think a lot of people, when they're writing this kind of thing, they think that, oh my God, I have never, like you said, I have never used this microscope before. I don't know how sophisticated the data can be. What we're looking for is not sophistication. What we're looking for is actually the fundamental variables that you would like to measure. It is either increase in intensity, increase in number, or changes in color, or changes in position if something moves, and how fast they move. So you notice that the 4 or 5 variables that I have just said are very, very fundamental, very basic, but you have to nail those down. The rest we can help you. Does that answer your questions, Sunday?
Yes, perfect.
Keep it simple, keep it fundamental. The fancy stuff we can help you, but the foundation we cannot help you. You have to come in with the foundation. We can add fancy stuff to it later. Okay, good.
Prof,
right.
There is another question.
Yes.
From someone asking, can the hypothesis be more than one?
Yes. In fact, though the worst kind of hypothesis is a hypothesis that is like a huge paragraph, like 4, 5, or 6 different phenomena all roll into an all-encompassing hypothesis. That is the worst kind of hypothesis you can write. Hypothesis should be just one sentence. And if you are addressing 2 or 3 different questions, sometimes one central question may need 2 or 3 different hypotheses to tackle, and that is fine. Then you actually have specific aim 1, a mini hypothesis, followed by 2 or 3 experiments. And then your second specific aim has another mini hypothesis, followed by 2 or 3 specific aims. So those kind of combination actually keep your proposal very focused. As a reviewer, I would say, oh, okay, this makes sense. And in specific aim 1, the applicant has a very focused mini hypothesis. It makes sense. And I look at the experiment and I say, okay, these experiments are designed to address that mini hypothesis 1. Okay, score. And then I go to second specific aims, and you have another mini hypothesis followed by a set of experiments that challenge it. As a reviewer, I look at it, they all make sense, you score again. So this is how you start collecting points for your proposal. So if you write a very complicated hypothesis, and I cannot even put my finger on exactly what you are trying to address, then when I read your experiment, it becomes very complicated. Now I cannot draw the arrow back from your experiment to your hypothesis. That is when you lose a lot of points, okay? Does that answer the questions, hopefully? Is there any other questions in the chat box?
No, go ahead.
Okay, all right. So now we go to the next one, which is equally as important. But nothing is as important as your hypothesis and your specific aims. If those two don't connect, and if those two are not focused, and they are not rigorous, your entire proposal is tanked instantly. That section, if you don't write it well, your proposal is gone. If you write it well, then this particular section called experimental design will help support it, will give you even more points as the reviewer is reading, right? So the experimental design section should detail the design of your experiment. It should provide a brief outline of every single step of your experiment. You don't have to go into super detail, but we kind of need to know what you're trying to do in the experiment. You want to describe the experimental condition and how they'll be controlled, all right? And then describe your labeling approaches. What is the antibody that you will be using? If you're using a fluorescent microscope, what are the some of the fluorophores? That is very important. If you're using a GFP and combine it with an Alexa 488, that's a horrible combination because both of them are green. So that kind of thing makes no sense experimentally, or technically it makes no sense, and you're going to lose point on that. So make sure you spelled out the antibody, you spelled out the fluorophore, you spelled out the dye you will be using. Again, if you're not sure, the AMI staff can help you. And it is also good to let the AMI staff know whether or not the sample is going to be live. Is it going to be fixed? Live samples, they will have to, step in and do a little bit more logistical support. How you're going to transport your live cells, your live samples. And if you're imaging plants, plants usually is always involve a lot more steps when you cross border. The recording has good. All right. So, for example, you hypothesize that with the gene expression, this particular gene expression, your protein of interest has moved from the nucleus into the Golgi apparatus. What kind of resolution do you think you would need? Micron, high 100 of nanometers, millimeters. So you have to tell people what kind of resolution you will need. You may not be completely correct, but start with something. What kind of speed do you think you will need to capture the data that is required to give you the information to test the hypothesis? You will notice that we're going in reverse as we talk about in the flow chart at the beginning of this workshop. So now you are really nailing down all the parameters that you need to produce the required data that will give you the information that you will need to challenge the hypothesis. So the resolution, speed, the duration, how long are you going to image that live cells, for example, 30 minutes, an hour, 2 days, let people know. The time interval, are you going to capture a volume or an image every 30 seconds, every 5 minutes, every 1 minute? Let people know, because this is important. If you're imaging something for a very long time and very, very fast, and if you pick a fluorophores that will photo bleach very quickly, your proposal may sound good on paper, but it will actually never work in practice because your fluorophore is going to photo bleach very quickly. So again, tell people what kind of data will be required for you to generate the data information that can actually challenge the hypothesis. And again,
I can add one.
Yes.
I just want to reiterate one thing that Leong said is it's really about what you need, not necessarily what you want. Right? It might be really interesting to do super resolution imaging on your sample, but if that isn't required to test your hypothesis, don't put it in the proposal, right? It's really about what imaging parameters you actually need to challenge your hypothesis, not what you might want. You might not need to image every second or something like that. So really think about what parameters are necessary to answer your question, not necessarily what in an ideal world your perfect experiment would be.
Yeah, and you will also see in the example below, also the number of samples is important. If you come in with 5,000 slides, you're not going to finish imaging in a month. If you propose you're going to have 20 mice, I don't think Army Imaging Center has the resources to support so many mice, so many animals. So these kind of things are important. So let's go through this example together. It is quite detailed. I don't think you absolutely need that level of details, but we can discuss it together. So the example basically says samples of diseased intestine will be obtained from patient biopsies. I think everybody can write that if this is your project. The sample will be fixed with formaldehyde, embedded in paraffin, and then the section is about 50 micron thick. Fine. I think, again, everybody can write something like this. This is your sample. And each slice will be processed as standard and labeled with primary GOAT anti-CD4 antibody, followed by secondary rabbit anti-GOAT IgG, conjugated to Cy5. So now you see the antibody is mentioned, the thickness of the sample is mentioned, and the dye, Cy5, is also mentioned, right? And then the sample will be mounted on the cover slip using some mounting media. Mounting media we can discuss. If you didn't put it in, you're not going to be penalized by that. So I wouldn't worry about it. And you can also say that nucleus will be stained by DAPI. Okay, good, because now you can actually count the number of cells, and you can count CD4 positive cells, right? So this is all kind of important. And then for acquisition, the applicants say they're going to bring in 40 slices, fine, of disease and control. So that's 80 slides. That's a lot of samples. Do you really need that many? How many do you think is required for statistical analysis? This is up to you. You guys should be good enough with statistics to design your experiments anyway. And so, but we can always provide a little bit of help. Okay. And we will be using a 20x magnification, something like that. So this is sufficient details for the reviewer to gauge whether or not your experiments design, the microscope and the Army Imaging Center can actually cater to your project, and whether or not your experiment is designed in such a way that it will provide the information that you will need to do image analysis. Okay. Any question at this point? Anybody else from AIC or Army wants to chime in? Okay. Now, this is the time when you
I was reaching for the thing. This is important too. Sometimes it's helpful to put a table in on which type of samples are going to be your control, what your imaging plan is for that, and your experimental sets, so that the reviewers have a handy.
Very, very good idea, Heather. That is a very good idea. A table sometimes beats 1,000 words. You can describe it in 5 paragraphs, and sometimes the reviewer will still get confused. But if you create a table, these columns are all the slides that are you want to test. These columns are all the slides that are controls. And what are the dyes that are involved? What are the stains that are involved? And it will save you a lot of words to discuss something more important. A table actually makes everything clearer and save words, and it also gives you yourself an overview of whether or not your experiment is actually well planned, because it actually helps you visualize your entire project that you'll be doing in Cape Town. Okay.
If I can chime in, it would also be helpful in a table to arrange your experiments in a priority order. Like there are some experiments which are necessary to address the hypothesis, so put them as the highest priority. And there are some other experiments which might help your hypothesis but are not truly necessary. If you have time, you can do it. So it's very necessary to have a priority order in your experiments, because it's not possible to do all the possible experiments within the limited time you will be there.
Thank you, Hari. Another good point. Any other question or comments? All right.
I'll just kind of reiterate. I mean, the table is a wonderful idea. It helps you prioritize it. It also helps you think about the scope. If you can use your table as a way to make a timeline, you know, given what you know and given what you learned from your technical consultation, you can help then make sure that the scope of your project can fit in the time frame that you're given.
Thank you. And as I mentioned below, this is where, if you're not sure, request consultation. Do not write your proposal if you're not sure what you're supposed to be writing. Some of these will require you to know a little bit about the microscope. So this is a good time for you to, this comes back actually more towards Sunday's original question. Sunday, your question is actually more relevant here. Is the expected outcome is very scientific, and as a scientist, you actually should know your expected outcome and how it will challenge your hypothesis. What you may not know is what are the capacity of the microscope. This is where you actually should absolutely request consultation with the Army staff, and this is where they can help you. Michael and Karen can help you with that. And then your quantitative measurements, and this is where a lot of biologists tend to get stuck because to many biologists, not just in Africa, worldwide, this is a worldwide phenomenon, that people still don't think of microscope as a quantitative tools. Besides getting an image, what exactly can a microscope do as a quantitative measurement technique? In fact, microscope is a highly quantitative tools. And this is actually, in fact, to add to Michael's original challenge, is I want to challenge everyone who is listening to start thinking about microscopy as a quantitative approach. It's not just a phenomenological approach anymore, okay? So as a quantitative tools, the only thing quantitative tools can do is to measure variables. That is what a quantitative tool does. So you have to think about what experimental variables that can be measured from your project that can challenge your hypothesis, right? So the measured variables must absolutely test the hypothesis. And you want to avoid, again, very vague words that doesn't challenge your hypothesis and frankly cannot be measured. Avoid words like spatial temporal dynamics. No microscope can really measure spatial temporal dynamics. It can follow spatial temporal dynamics. It cannot measure spatial temporal dynamics, because what is the unit, measurement units for spatial temporal dynamics? It doesn't exist. I want to characterize something. Well, that is not a measurement. What is the unit for characterization? No. If you cannot even come up with a unit, like intensity value over a unit area, or the velocity, or cell density, which is number of cells per unit area or per unit volume. If you cannot even put a unit on it, that is not a variable that you can measure. So always think in terms of unit, then it will help guide you to the right quantitative measurements. Another very important thing, and this is where the Army staff plays a huge role. You do not need to know how to analyze an image. You may not know how to use image analysis software. That is fine. Army can help, but you must let the Army staff know what you want to measure. They are not going to tell you what to measure, because what to analyze, what to measure is. Make sure that you can do that. Okay, can everybody hear me okay still? I have an error message. Okay, all right. So if you look at the examples below, this is a pretty straightforward quantitative measurement paragraph. The DAPI signal intensity will be used to segment the nuclei, and then which will be used to as a marker to identify all the cells, right? So DAPI help you identify all the cells. And then the nuclei within the defined distance of the threshold of the Sci5 signal will be designated as CD8 positive cells, right? So they stain CD8 as well, in addition to DAPI. So DAPI gives you all the cells, and then CD8 positive cells you can measure with CD8 intensity. And then thereafter, the proportion of CD8 positive cells relative to the total cell number will be calculated per square millimeter and compare between tissue locations and various disease states. So these are highly quantitative statement that you can use. And these are the things that you should be able to come up with. If you read it very carefully, nothing fancy is in that short paragraph. Everything is something that as a scientist, you should be able to come up with, right? Any questions at this point? I saw that there are a few things added to the chat box. Nothing? Okay. Good.
Leong, if I'll add one thing, one way that I've found helpful for myself to think about what kind of quantitative metric I want is, envision that you finished your experiment and you're writing your paper and you're putting together the figures, right? You have, some bar graph that's going to compare your control and your treatment. What's your bar graph showing? What's on that axis that you're comparing a change in, right? It's not going to be characterization or spatiotemporal dynamics. It's going to be something specific like cell density or intensity or something like that. So envision you're putting together that figure, and what are you comparing actually between your two conditions?
And Chad brings up a very important word that I think deserves a little bit more mention: compare. A lot of biology experiments are really only meaningful as comparisons. So for example, between a treatment and a control, or a wild type and a mutant. So I think in general, projects that are comparing values between different kinds of samples tend to be the strongest. That doesn't mean that's always the case, but that is very often the case. So when you're designing your experiment in thinking about how you test your hypothesis, don't just think about one sample. Think about something in a control or some kind of comparison that you can make that really, really solidifies that. And a lot of times that comes out when we talk about quantitative measurements, and you start thinking about the actual quantitative numbers that you're going to pull out of these, out of your data.
Good. Thank you. Are there any questions in the chat box? No?
No.
All right. All right, again, if you're not sure, request consultation, right? Stephanie raised her hand. Stephanie.
Yeah, thank you very much, and good day. It's an afternoon here, and thank you for the presentation. Now, my question is, when you said from the slide, AMI can help to analyze the image, but of course, they must know what, what we want to analyze. In that case, that means that there will be would there be a kind of an MOU between AMI and the researcher? That's my question.
So the MOU for what? What do you mean by MOU?
What I mean by memorandum of understanding is, is it in terms of partnership? I mean, or just to analyze, and would they be acknowledged, or they will be part of the research work? That's just what I want to know.
Oh my God, let's address that.
I can answer to that. Yes, there will be an MOU set up between AMI and all visitors. Most of the work and support provided by AMI to researchers will be of the kind that requires acknowledgement, and that acknowledgement will be absolutely essential for the ongoing operation. So that if researchers fail to sufficiently acknowledge in outputs, we are unlikely to allow you to come back to the center. But depending on the input required, so the time and level of complexity, we can discuss whether that is a collaboration warranting co-authorship or just acknowledgement, but that is something that can be discussed with each visitor. At every point in the project, the visitor owns the data, but part of the MOU is that there is an expectation that the data will be made available on a public database, open access to the world after a few years after your visit. And that is in line with the expectations from our funders. Does that answer your question? Or is there more that you want to know about it?
Yeah. Thank you very much. It's just that I was having some calls coming in and I couldn't what you said about actually there's always terms of whatever we want to agree on, you know. So that wouldn't be an issue, but I just wanted to know whether it's just to help to analyze, then the researcher can go on to replicate, or just that AMI will be fully involved in the whole research, you know, leading to even the image analysis, or at the point of just the image analysis, AMI will just come in. Yes.
So maybe, Karen, go ahead, and then I'll try to.
Sure. AMI will be as involved as we are needed to be involved. If we have visitors who come in and are confident with microscopy and just need training on a microscope and don't need help with analysis, that we train them on the microscopes. Obviously, we've screened the project, so then you require acknowledgement. If you come in and you need training on the microscope, help with the imaging, help with analysis, help with data interpretation, then we will provide that level of support. The more support required, we are open to, that will be discussion on a per visit basis, whether that is in a collaboration or just a service provided, but we are able to provide support as needed.
So Stephanie, let me also chime in, is a lot of the image analysis, especially if this is a fixed slide, it can be easily learned. And so when you're at the AMI Imaging Center, you will actually get to learn a lot of the software. So after a few rounds of analysis, you can actually do it at home. And most of the software now, even completely free open source software, are extremely powerful. Actually, it is a good skill for you to learn. So what I think the first and foremost responsibility of the AMI staff is to train you to be as self-sufficient as possible and as independent as possible. Certainly, there will be a lot of training and teaching involved while you're there, but I would say that 95% of projects that go through AMI, the visitor can learn how to do image analysis on free software. So that makes it a lot easier. Does that make sense?
Yeah, thanks. I'll reiterate that quickly.
Yeah, it does. But it does, but I have a lot of issues with, because I am opportune to know about and to use CUPAD, but up to now, we're still having some challenges, which actually got me interested in this application. In live imaging. You know, when you generate data, we were using, at some point, I was made to use the traditional method, you know, microscopic taking at different times, but I am still not able to use, I don't know if you know about CUPAD, I know you may know about CUPAD, but even to use it to detect cells, there is a whole lot of problem. maybe, I don't know, I'm not with my laptop. I will have shared some of the images because I'm in a group and I've shared the question, but I couldn't get a response. And another challenge also we're having in this part of the country or the part of the world is developing even the image itself. I mean, the staining, the protocol, I mean, is very poor. I don't know how to, is unfortunate and is appalling to even talk about. But because if you don't get a good stain image, I mean, of course, every other thing is affected.
Yes.
So it's
yes.
it's a
Stephanie, why don't you talk to the Army staff, and then I'll be happy to chat with you as well. It'll be nice to see what kind of challenges you're facing, and this is where consultation really comes in, right? We'll be certainly happy to do work with you. Okay.
Yeah. Please do schedule a consultation with us, Stephanie.
Thank you very much, Stephanie, for bringing up those points. Those are very good to know. All right. Any other question? All right. Good. And then justification, and this is something that Karen, feel free to chime in. So the justification of the of why you want to come to the Army Imaging Center. This is actually a section that should be quite easy to write for most scientists working in Africa. And the reason why Army Imaging Center was created in the first place is to actually give you the necessary resources. So it will be nice and helpful if you can discuss a little bit about the technical aspects of the instruments that you absolutely need. For example, you can say my university have 1 or 2 instruments, but none of them have the resolution or the sensitivity or the speed, or my university have no microscope whatsoever. And we absolutely need to address these questions, and this is why we need to come to Army Imaging Center. And that is fine. I'll leave it up to you to discuss what the challenges that you are facing. And this particular section will not be scored very heavily. So, because that's the whole point of Army Imaging Center. So as long as we know a little bit about your situation, this is fine. Karen, I'm not sure if there is anything specific that you would like to add. Other than that, I think we can move on. I think most people on the Zoom in this section pretty well.
Yeah, I think that is fine. I think that there's a specification that just think about the rationale why you're wanting to use the specific instrument you're wanting to use. And then obviously the question is, where else can you access that instrument? And if there is nowhere else at your institution or nearby, then that is justification to visit the imaging center. Please do take a look at the resources available on the ABIC website to confirm if there really is nothing nearby you, because that is one factor that we are going to be looking at in review. If you're applying to use the confocal and you're in Accra, we know that there is a confocal in Accra. We're going to tell you to go use that one, unless you need a feature that our confocal has that the one in Accra does not have at Accra, for example. But aside from that, I think I agree, Leon, this should be fairly straightforward.
Okay, thank you, Karen. Those are all good points. There's something in the chat. Anything somebody wants to read out?
No, nothing.
It's just Ame saying that we're technically got 10 minutes left here.
Okay, I'm almost done. So this is pretty much the last slide, and this is the impact. Impact is something for you to discuss why this work is important. Now, you can certainly write that, well, if I have these images, I can cure cancer, but that's not the point. The point is articulate how this project can really affect your research direction and your lab. You can say that, I rarely use microscopy as a tool, but I know that my future research direction will rely heavily on this. This will give me an great opportunity for me to explore how microscopy can help my research. And that is perfectly fine. This is the kind of impact statement we're looking for, right? And so you can say, how the exposure to advanced microscopy changed the outlook and the direction of your research. And will you, for example, use the data collected in a manuscript that you'll be preparing? And will you be able to translate the knowledge that you learn at the Army Imaging Center to your colleagues, to your students? It will be really nice to talk about that, because we certainly want to continue to expand the use of microscopy in Africa. How will access to this microscope resource change the way you apply for funding in your home institution, in your home country? And especially if you are a professor, can the Army Imaging Center help you reconsider, rethink how the research infrastructure back home can be reconfigured, for example? So any of these things that is not completely or directly related to your project is what we are interested in. So this is actually quite important, because at the end of the funding period, when we are writing a progress report, these kind of statements will help Army secure future fundings. And so do not just, skim over it. Give it some thought. Consider how what you write can help our Army secure future fundings. So think about it a little bit more before you write something. And I believe this is the last slide. All right, I am going to unshare my screen, and we can continue to discuss until the time is up, I guess.
Okay, thank you, Prof. So the floor is open to everyone who has a quick question, a quick comment.
Sunday has his hands up.
Thank you, Leon, for the presentation. My question is about timing. From the time your proposal is accepted what is the duration of time to when you go to AMI to carry it out? Is it as soon as you are ready, or when the AMI will determine when you come for the work?
Karen?
Good question, Sunday. So this will be planned together with us here at the Army Center. So we will schedule your visit, especially for those of you coming from outside of South Africa. We're very aware that you need to get visas, and that can take a while. So we will plan your visit months in advance. Firstly, so that you can do your visa application, and secondly, so that you can make sure that you have your samples ready to go. You've done all the validations and controls that might still be outstanding, that sort of thing. And obviously, it's dependent on both our availability, because we'll be scheduling lots of visits, and your availability, because you've also got stuff going on. Does that answer your question, Sunday?
Yes, thank you. Thank you.
I guess for those, if we've still got anyone online from within South Africa, obviously visa are not a limitation. So we've got more flexibility in scheduling your visits on a shorter timescale if you're ready.
Okay, thank you, Karen. So just a quick reminder of our ABIC online community update meeting. I remind you it will be on April 20th from 1 p.m. to 2. So you are free to go online, register, and join us on April 20th. So any other comments?
Yeah, there are two more questions. Stephanie asked, how is Army related with ABIC? And vice versa. That's question number one. Question number two from Yvonne at Uganda. Karen, you might want to answer Yvonne's question first. Will the center provide consumables, such cells, for instance?
So in terms of costs of experiments, such as consumables, yes, we will cover the costs of the base costs of your experiments, including consumables. Cells, you obviously have to let us know what cells you need, and if they're a highly specialized line, or if they're available via ATTC, for example, we can order them in. If you're doing a screening, like an siRNA screen, for example, and needing to image a large number of plates with very expensive reagents, we cannot cover the full cost of an experiment like that. We will discuss with you a budget.
Yeah, I think it will be case by case basis.
Yeah,
right?
that is the case by case discussion. But base cost of experiments, yes, we can cover, including consumables and cell culture.
So may I answer Stephanie's question?
Go for it.
I'll answer it in a very short way, and then Karen can expand it. So think of ABIC as a community. It is a community outreach program. This is how the Africa community work together around microscopy as the central theme. So there is, instrument sharing thing, and then education. There is training. There is also awareness of other funding opportunities and everything, or a proposal writing workshop, for example, that AMI is putting together. ABIC is putting together. Now, think of AMI as an infrastructure. AMI is truly a capacity strengthening initiative. It certainly has a little bit of a community element to it, but the most important thing is to make microscopy available to African scientists, and is to also bring expertise to microscopy expertise to Africa through various approaches. So one of them is truly a capacity strengthening initiative, which is AMI, and then the other one is a community initiative, which is ABIC.
Nice, thank you, Prof. So yeah, just 1 minute ahead.
All right.
I would like to
Thank you.
To maybe let the floor to Prof before we leave. And thanks to the whole team.
Yeah, and let me chime in one last thing because I saw the word clinics there. So after all the tips that you have learned today, hopefully it is helpful. We will be organizing clinics. Basically, we will take your proposal that you have written, and maybe just 1 or 2 proposals at a time. During the clinic session, we will help dissect what is good and what can be improved during the clinic, so that you actually get feedback before you submit the proposal. And along the way, you also get to learn how to write better proposal. And that is what the clinics is all about. Workshop is just teaching. Clinics is to take your actual proposal draft and dissect it and help you write it better before you submit.
So I will follow up from that. A number of you have asked about materials and recordings for this meeting. As Leon mentioned, they will be available. We will send you all a follow-up email after this workshop with the recording of this, as well as the information on how to sign up for one of those clinics that Leon was mentioned.
Thank you, everyone.
You'll look out for an email from MA about that.
And everything that the slide, I realized that there are a few typos, so I'll fix it, and then I will share it with Amy. And then also the paper that I mentioned will be shared as well, and you guys can just distribute it after I've shared.
Okay.
All right. Thank you.
Thank you, everyone.